Okay, cool.
All right. So yeah, I have a great pleasure to introduce you, Erik Garrison, that is our first guest lecturer. And just to give you some context, Erik is currently an assistant professor at Tennessee University, and he has a PhD in genomics from Cambridge University. And his mentor was Richard Darwin, as was one of really influential people in computational genomics, bioinformatics. Erik, has done a lot of things, so I'm really excited that, he's here with us today. He was part of the 1000 Genome Project. He wrote like a lot of softwares that are heavily used for sequence alignment, variant calling. And today, we'll talk about this set of methods that it developed to essentially think about how should we store genomes, collection genomes. I will talk about these pangenomes and as well long reads, and he's really a pioneer on this field. So I think there is no one better than him to talk about these topics. And I just a brief, and also it was really kind, to join us today. I still remember the first time I met Erik. I was a biology of genomes. I saw his talk and said, wow, I mean, these methods will change everything. And then we started to chat and to collaborate. And one thing I want to say, these are methods that will have a tremendous impact on many of the downstream analysis that we do. So you can envision there will be a lot of tools that will use this data structure. Actually, we wrote a tool with Erik about characterization of TF binding, but these are just a few examples. So I don't want to take more time. Erik, thanks again for joining us today. And I will leave the stage to you.
Thanks. Thank you, Luca. Thanks for introduction. Thanks for having me. It's a sort of unique pleasure to come and virtually, unfortunately, but to come and give a talk, because I was actually an undergraduate at Harvard, and I studied social sciences, social studies, which you guys probably know what it is. And in fact, I got into genomics after graduating, because I ended up, this story is a bit funny, but I was really interested in finding places where small amounts of work could have very large impact, and small amounts of resources as well could have very large impact. And I thought that open source software was a domain that would be interested in. And so I got involved in the free and open source software community and free culture communities at Harvard, and kind of transitioned into this more technical space than I'd been in. I was working in a sort of philosophical dimension in my thesis, my undergraduate thesis in social studies. And I ended up working in the lab of George Church when they were developing the Pauline sequencing system, the first kind of attempt to make a commercialized, in fact, it was also meant to be open source, a commercialized sequencing platform based on short read sequencing. And so that threw me into the end of bioengineering, where you start to think about how biomolecules interact with each other and interact with larger systems, and how we get observations out of them. And basically had this foundation for the I guess the trajectory that I'll be describing here. It starts with looking at DNA and trying to understand how what that represents. And then once you understand the genomes it represent, trying to understand how they relate to each other. And it's like, you're like, okay, that's just bioinformatics. But that's a bit of my backstory. And so as of pretty recently, I'm physically based in Memphis. Before I was in Europe for many years, actually, starting a family and also doing graduate school. And anyway, we're living in a virtual world, and so that's kind of normal. So without further ado, the background for this is kind of basic. When we work in bioinformatics, we want to look at genomes, and in fact, it's not really just the genomes we're interested in. We really would like the whole tree of life. Like we'd like to imagine the whole tree of life and look at all parts of it, the whole evolution of every little bit of sequence, how it changed, and so on. That would be really helpful to see. And so here, this is a really horrible schematic that just says, hey, we get these genomes. Imagine their genomes are kind of, DNA is a bit like has a kind of crystalline aspect, right? Because it's like over time, it's getting copied exactly. And so this sort of showing you that aspect. And then sometimes there are differences that arise. Those cause variations in this sort of phylogenetic tree as we go through variation. And so the red and green represent variations. And the standard in the field, in the fields that are interacting with genomes right now and working on resequencing, is to take a single reference genome from this tree and then relate everything to that reference. And so that's kind of a genomic model. So a genome, or the genome, depending on how important you think it is the center of your analysis. And so here, if that genome is R, then A, B, C, and D are new genomes. And we just can relate them to each other via that reference. So we align bits of sequence from them that we've obtained from sequencing. And then we can build up a model that incorporates them all. And to extend the model, it's pretty trivial because you just have to do a pairwise comparison with the reference. Some new data set, delta, you're going to compare it to the reference, right? This is super scalable, amazing. We can look at hundreds of thousands, we can look at millions of genomes this way. And there are companies and organizations that are doing this at a tremendous scale. And it's scalable because it's very parallel. You just have the reference, and you have a new sample, and you compare. But it kind of breaks down because, right. The process is this simple. There's literally a sequence file in what's called FASTA format, mostly just ATGC and N written out. And then you take short reads from some sequencer, and you do an alignment process, and you do some filtering on the alignments, and apply some models that let it see what are the small variants. And you get this output that's basically genotypes in the sample across sites relative to the reference. So where are the point mutations? Where are the small variations you can see in the reads? And this breaks down really fast, actually, in a way that's kind of funny, because even a single individual genome is not easily represented in the FASTA. So this is a haploid genome that we're working with. And when we find variants, we can't easily update it so that it incorporates them. We have to basically pick which one we add in. Like we can make we can sample a new reference, but it won't incorporate all the variation we know about. So we're stuck in this sort of place where the reference is a fixed entity, and differences from the reference are going to be hard to see because we don't have the sequence information that's required to contextualize new sequence observations. And so a solution to this is to make the reference system not a linear sequence, but some kind of encompassing model. In the cases I'll provide today, that's a graphical model. There's a graph, has a certain structure, and it lets us represent multiple variations, multiple sequences in the same context in a non-redundant way. There definitely are other ways to do this. And by no means do I want to exclude those. And I think that possibly, this particular solution is just part of the problem, and we'll end up with something much nicer and more generic in the future. But practically speaking, this is a well-established idea that's being refined into this particular topic, and it's been extremely useful in that context. So, so the gist of this, though, is that to make that kind of graph, as I'll show, you can't just compare things pairwise with the reference. It makes it, now you have to really think about all-to-all relationships of some type. And you can see that the number of relationships is going to grow very fast relative to the number of samples. And adding a new sample is now much harder because you have to relate it to many genomes that are, perhaps they're embedded in this kind of non-redundant structure. So it's easier to do that, but it is just practically harder. And there are all kinds of conceptual and technical challenges that are related to this that we've been resolving as we've built out this kind of space. So the model I'll be focusing on in a lot of the talk today, a lot of the discussion, is called a variation graph. And this is really meant to be a mathematical term that says, this is a sequence graph that has sequences attached to nodes. The nodes have identifiers. There are edges between the nodes. Those show you ways you can traverse the graph that would write genomes or sequences in the language represented by this graph. And then we also, and this is the key part, we also record the original sequences as they're transformed into the space. So every sequence we use to build the graph can also be written out as a series of node identifiers that it walks through, like a series of words. And so this is a very, it's a lossless kind of model, and it implicitly gives you all the mutual relationships between the sequences embedded inside of it. Maybe it's not clear exactly how that works, but by implicit, I mean things like red path is my reference, and I want to know the position of a given node in it, then I can go into that path and I can walk back to the beginning and count the bases that there are there. And I can do the same for this other one, right? And so on. So these paths also provide positional systems that let us project annotations and external information, other information that's related just to those sequences into the same model. And that's really nice because it lets us work with existing tools that work on single reference genomes or pairs of genomes, or annotate an assembly. They don't have to think about the graph because having embedded the linear sequences in the graph gives us a way to convert them and put them inside of that space. So, and then the last part, this is kind of an academic question. If I didn't keep these paths around, the number of sequences that this language model, which is kind of equivalent to a regular language, the number of sequences it can represent is very, very large. It kind of grows exponentially as we go through furcations. And if it's memoryless, then we can't learn things about the correlations between different nodes in it. And that makes it really hard to do all kinds of stuff. So we just have to keep information about representative sequences or sequences around. The combination of the sequence graph and those paths makes a variation graph. And they're really meant to be an answer to this key problem in bioinformatics, which is how do we represent both sequences and any kind of variation between them? So not just sequences and small variants that are kind of linear bubbles, even like the things we're seeing here, but also structural variants that might involve inversion of sequences, repetition of sequences, connection between different chromosomes, connection between different sequences, translocations, and these kinds of things. So this model can encompass all of that. And really anything that a piece of DNA can do in evolution is going to be represented in a direct way in this model, and in a way that we can look at, interrogate, and build algorithms on top of to work with. And I really think that this is obvious enough that people just assume that's how we're doing it. Like how do we bioinformaticians work on genomes, right? And to make this point, I'm going to pull something out of the social web, and it's kind of a funny vignette, but I wish I had more examples. Basically, this idea keeps coming up in the popular press. Actually, there's just been a new article basically saying the same thing as these, but it's in The Guardian, and interviews folks that I've been collaborating with, among others. But the gist of it is like, okay, we made the genome assembly, one human genome. Then we took like thousands of genomes and we align little sequences against that, and we can find all the different they have. And we find, of course, not everyone's the same. We know that to be the case. And maybe there's some mathematical approaches we can use, like the ones I'm showing, that'll make it easy to model that. And so there are a few popular science articles that come out. As I was a PhD student, I was collaborating with a group that was mentioned in one of them, and the second one by Carl Zimmer. And it has these same kind of funny visualizations that we're showing in previous slides. We got those into the popular science press. So it has, actually, this is a really silly aside, but basically there's a hash of the name of the sequence is used to make a color and emoji. And we did this so we could have hundreds or thousands of combinations for debugging purposes. So that's the reason there are emojis in these renderings. This comes up on Hacker News, and that's where this vignette comes from, is that Whitney Land says that something doesn't add up because it must be that people were always smart enough to know that this would be a problem. We always had enough information to know this would be a problem. So why didn't they build the right tooling from the beginning? I actually know the answer to this because I talked to folks, including Richard Durbin and David Hausler, who were involved in the conversations as part of the GRC that was making the first reference. And that data actually comes from a mixture of different donors. And so the GRC human reference, the standard reference, it's mostly derived from one individual who actually happens to be admixed African and European, but a lot of it comes from others. And so you have this kind of variegated, complex pattern that actually is graphical. And so they did assemble the graph and they wanted to release it. There were folks in the project who wanted to release it. And in the end, they said, no, this is too complicated. The group decided that they would just get one assembly. And that was the most important thing to focus on. And actually, you can see evidence of this in the fact that on UTHSC, there's reference to something called the golden path. And that golden path is the reference. And in fact, it's walking through the same kind of graph that we're describing here. But they kind of got rid of the graph. So yeah, so these are not how we've been doing things, but how we can do them. And it will take a lot of update to many kinds of tools and methods to make them usable. But they give us some advantages. For one, that can provide us a very complete and simple model of the relationship of many genomes. And by simple, I mean that graph can correspond to a matrix representation or a vector space representation. Here it's just shown as a binary matrix of the paths below the graph. And every one of these vectors encodes almost everything you need to know about variation in that individual. And so we can work directly on those vectors and just forget about the sequences at the level of applying statistical tests and mathematical analyses to the genomes. I think that actually is really simplifying and could be really, really helpful in the future because, and it is helpful already now for our work, but if other groups start to adopt this kind of thinking, it'll be helpful for the basic fact that it makes all variation the same in the sense that there's not a difference between SNPs and structural variants or SNPs and indels. Everything is just coverage or presence or absence across this kind of pangenome space. And that's something we haven't been able to do easily because we have all these problems about how do we relate structural variants and SNPs because they happen at different scales and we think about them in different ways. But this is at least a model that will let us put them all together. Also, I want to point this out, that this is an idea that's basic enough that it's not new, and it came up in another context where human beings had to understand variation of sequences, which is in archival research. So this comes out in, I think this is published, 2009. So years before I started this stuff with variation graph. They call this a variant graph. The nodes and edges are reversed, but conceptually it's pretty much the same thing. Here we've got 9 versions of a poem about the Roman countryside that basically let us look at what parts of those versions were conserved and what were varying. And we see that this is discussing the arc of the bow of violin. The word measure is obviously very important. Reference to silence in the country, those are key things. And we can see this in a way that's very easy, but it would be very hard if we had to pairwise align 9 versions of the poem to each other. So it's an idea that's been out there and is I think actively being used in other fields. And so to implement all these concepts and try to make good on these ideas, the original vision was like, everywhere we have a reference in bioinformatics, we're doing basic bioinformatics stuff with it, we're going to put a graph there and a variation graph and implement these ideas and as a research delivery vehicle to make code that people could play with and build on. We developed VG, Variation Graph Toolkit. And this schematic on the right is actually from a review paper that came out around the time we were starting VG. And So it comes with some of the ideas from the developers of VG, but also from collaborators who are thinking about similar kinds of things. And we point out that this is like the kind of network of stuff you might want to do when you have this pangenome model. You can imagine building the pangenome from a reference plus short reads, or a bunch of assemblies, assembled sequences. We can somehow construct it. We're typically doing that with a VCF file, like a variant call file, maybe with phase haplotypes. So it's a reference-based approach was typically used. But then we get this pangenome that locally can represent all the variation, like the ways I was showing you in previous figures. Now, with a new individual, we can align their data into this pangenome, and we can do variant calling and phasing. And then a cool thing is that we can update this, which is not really possible in a kind of standard approach that's based on the single reference. So this ability to add more information in, that's one of the things we win. And we also win this possibility of visualizing and interacting with the system in ways that were not possible previously. And so this toolkit then was used in a series of papers that showed first it was possible, and then what kinds of specific applications was it helpful for. And I'll just go through those here so you get some lay of the land. So this is the first one, the variation graph toolkit. It's kind of the original implementation. I did this as a PhD student. And I think that probably the most key result in this paper is this figure, which shows that when you have a heterozygous variant in the sample, so in this case, it's just this validation sample that's been used by NIST to establish standards in resequencing technology. It's called HD002. So if you have that sample, NIST is telling us the truth of the genotype state and the alleles. And so we can use that, and we can ask, at the sites that are actually heterozygous, how well do we observe the alternative non-reference allele in different conditions? So if we align to just the reference and do variant calling with freebase, which is actually variant caller I developed. So BWA and freebase, this is blue line here. And what this is showing us is that at a SNP, the delta to the reference is 0. If we go positive, the delta versus the reference is it's basically an insertion relative to the reference, and negative is deletion relative to the reference. And that's how we've organized the data. So for SNPs, there's kind of no, you see exactly half and half. 150 base pair, paired end read, you're going to be able to map through a SNP. Not a problem. But what begins to happen is as you see larger insertions and deletions, you begin to miss the alternate allele, the non-reference allele that has this delta in it. And that's quite profound as you get close to a third of a read length, so about 40-50 base pairs. This is also the case. Even if you use VG, but the variant isn't in the graph, you also have reference bias. And that's the case for some fraction of the variance. Pretty small fraction, which you can see by the error bars on this green plot, but it's there, and it's kind of similar to the pattern we see when we work on the reference. And this is not the case if the 1,000 Genomes project called a variant, so it's in our graph, then we align to both alleles as if they're exactly the same. And you can see that because this red line is always at 50%. So at heterozygotes, we always see half and half, right? And so this shows this removes bias in real data. And so we do this we have an application on ancient DNA, because in that case, you have short reads, they have high rates of error. You know what variants you're interested in many cases, or you have difficulty calling new ones. And so you can totally imagine putting them in a graph, and then you remove ancient DNA bias that you typically have. You have very strong reference bias, which actually this has not actually been applied in practice very often because of cost, but really because of community practice in ancient DNA. I can get into that a little bit, but the gist of it is that if you have bias toward the reference, it's going to make it really hard for you to look at evolutionary trajectories over ancient DNA, because so much of what you're seeing is just coming from reference bias. There are all kinds of statistical methods that try to deal with this, but none of them actually solves it completely, except putting the variants you're interested in the reference. And so here we have examples of what happens at the CCR5 delta mutation, so it's called. This is a big indel deletion. If you have the deletion, if you're heterozygous for it, you are more resistant to various kinds of infections, specifically bubonic plague, and it's also been shown to matter for HIV. And if you want to go to ancient samples and look for the presence of this mutation, you have to do some custom pipeline, because if you just use BWA align to map the reads to the reference, you cannot see any observation of that indel. It's just not designed to find a 32 base pair indel and a 50 to 60 base pair read. But if you put it in the reference and you map with VG, then this is actually not against the graph, right? It's been, we call it surjected back to the reference. So you can kind of observe it in IGV, but you can always see this when it's present. Even when there's very few reads, we're actually able to get some information about it. And so that's promising, but the field doesn't like this very much, because there's always an attempt to kind of exploit the data for the most possibilities. And so they only release reference aligned reads as a standard, which is really, I think it's abhorrent. I don't mean to, I mean, I really think it's bad. I wish that could change. One of the reasons is that the idea is the unaligned reads, maybe there's some metagenomic information about the microbiome of the individuals or of pathogens. So that needs to get exploited too. I think it just should be a requirement of funding agencies that they're releasing all the data so we can actually work with it. So then this is work with Luca, and we thought, okay, well, Luca really drove this idea that basically, just as we were seeing for these poems that we could compare to each other, it can really help human understanding to see these graphs. And so we wanted to kind of like, well, the long-term vision is like building the graphs to represent the motifs. But the first thing we did was actually just to use this augmented reference of the pangenome to help us scan for motifs and find things that weren't in the reference. And so this is a CTCF motif. And basically, we're just scanning this across all the haplotypes that are embedded in the graph. We have a way of enumerating those, a way of showing which ones exist in some samples, which ones don't, which ones are like putative recombinants. And then we can look at cases where the non-reference allele actually is a much stronger match. And so the non-reference alleles like basically would be associated with the motif of being a binding site, but the reference ones wouldn't. And so we did this kind of study to prove the concept in effect. And then I was talking a lot about structural variants, but until this paper, we hadn't really been careful about evaluating how well we could study them with the VG toolkit. And so Glenn and others, in collaboration with me, I think it was still a postdoc at this point. It was a postdoc for about a year with Benedict Hatton. And excuse me one second. Well, anyway, we're playing with VG, and we're like, okay, let's see how well we can do at detecting structural variants. And one problem was basically that at the time of this paper, we didn't have a lot of whole genome assemblies that were high enough quality and haplotype resolved that we could actually look at structural variants in them. And so all the data we had was from short read data sets, from things like from lower quality assemblies from a project called the Human Structural Variation Consortium, which is like an offshoot of the 1,000 Genomes that keeps looking at structural variants. But we had some nice examples of how this could help and how we actually were able to get genotypes reliably for structural variants that were in the graph. And so I've heard this is actually used in some production pipelines now as a way to get SV calls. So this is a deletion of an exon. It's from this HDSVC data set, and we can genotype it correctly. And you can see why, because if we map the reads, these are reads in red and blue across the graph. The graph is here on the bottom. Then you can see we can map across the deletion. Whereas if we map against GRCH38, you get this kind of mess. And what this is showing us is what's called soft clipping, that basically the reads don't completely align. And that this is a display that IGV uses to show that happening. So we can see that something's going on, but we can't see what it is. If we have a good prior, then we can see clearly what's up and genotype it. And so then this is the most recent extension of VG, is to not just make it work, make it unbiased, but to do that and make it fast and scalable, which the original version was not. It's something like 10 times. The original version is like 10 times slower than BWA, which is a kind of standard and not super, super cheap thing to run. So I think that really inhibited its utilization. So we tried to resolve that by rebuilding the alignment approach. So I didn't get into the alignment approach used in VGMap, but in effect, it enumerates all the possible k-mers in the graph, and it has a kind of index that looks us look up really long k-mers of hundreds of base pairs, and so we can exactly match the read sequences to that, and then we have this sort of post-alignment process. It's kind of similar to what we're doing here in VG-Giraffe, but this is designed to be fast. And so lots of things that were slow or harder took time. We were rewritten to be much quicker. And basically what you do is you get this index of a subset of k-mers, k-mer being a fixed length sequence, right, of length k, called minimizers. There's a selection criteria. Minimizer, it's sort of a term of art, but it just means that we are picking a k-mer that maybe has the minimum value in a given window among all other k-mers. And so we can pick one out of every 10, one out of every 50. This makes it much simpler to work with. It's a sparsification process. And that's used in other techniques like Minimap2, which you might be familiar with. It's often used for long read mapping. It uses this kind of technique. So you can take a read and you take this haplotype index, the variation graph, and then match across that. And then there's a distance index that it kind of assumes that the graph is mostly linear with small bubbles in it, which is pretty much true in the chromatic regions of genomes. And so then you can do this chaining based on a metric across that efficiently. There's a kind of greedy extension. And then if none of that completes perfectly, like if we can't just perfectly kind of match the read to the existing haplotypes, which usually happens in almost all cases, then we'll go and actually do pairwise alignment with the graph. And just as a sidebar, I know you guys aren't going deep into the algorithmic details of some of these things, but pairwise alignment, that's when you take two sequences and you describe the transformation from one into the other. And this is just to say, this figure is just to say that that generalizes easily to graphs and sequences or graphs and graphs. And so we use these kind of standard techniques. This one's called partial order alignment. We'll apply those to actually do the alignment against the graph. And so what's going on here is that it's showing in this very funny, like late 90s kind of schematic, that there's a bifurcation in the graph. And this kind of corresponds to like as if the matrices that you're working across are pasted together in this funny way. And so you have these different alternatives that appear when you're considering like where should the alignment go through that space of possible relationships. And so the method, it's not perfectly accurate, but it's accurate enough. And what I mean is that this pink line, that's the VGMap algorithm, the original one that I showed from 2018. And these are ROC curves. So the closer that these curves get higher to this upper left corner, the more specific and sensitive they are. The sensitivity is on the y-axis, and we have actually have a log scaled x-axis because that's the way the error rate goes. So you can see that this pink line is like better than VG-Giraffe, but VG-Giraffe is very, very close. And in fact, it tends to be better than methods that are based on just a linear reference like BWMM and Minimap2. And it's really, really fast. It's really fast, faster even than BWMM, which, like I said, is the standard in the field. And if you can organize its indexing in certain ways, then it can be really, really quick. And it does this a little bit by trading time for memory. So it uses a ton of memory, but on modern systems, it's not really an issue. A lot of systems have hundreds of gigabytes of memory. Even the laptop I'm on is 32. So this kind of issue is something that can be exploited also to speed up the method. And it improves variant calling in a way that's pretty awesome. Like it gets some of the best results that were ever seen by Google's deep variant team. They work on variant calling using machine learning, and they basically we do alignment and then this process to project the reads back to the reference, and that results in the opportunity to do variant calling. And these are the same kinds of ROC curves, but now for the variant calls, and they show that Giraffe is pretty much on top of all the other alternatives, including BWMM. And it's better. Actually, interestingly, it's better than VGMap, even though VGMap appears to have better read placement accuracy. And then they also we replicate the result from the VG paper that shows this kind of pattern of reference bias for BWMM, but not for the VG-based tools. And finally, this is in C. This also works for structural variants. So we have a truth set for structural variants, and we can compare to that and do a good job genotyping with it. Okay, and then so this is all great. Technically, it's very nerdy, and we're pushing, like, if you look, actually the numbers are pretty hilarious. It's like you go from 99.57 F-score to 99.61, right? It's not really inspiring, but this is what's cool. Because we can do accurate genotyping SVs, now we can look at the population genetics of them across all of these thousands of genomes that we have available in short reads. And we see that they really match what's happening for SNPs, which shouldn't have, it's not really surprising, but it's a confirmation we needed to see in a clear way. And that's what this tool has allowed us to provide. So, and what you see is this typical simplex where the African populations are over here, and then there's European ones, and the East Asian ones are all kind of diverged. A lot of, obviously the PCA has to do with the data you put in. I think as we get more African genomes, it might be a little bit more centered on the African component, but that's at least how we've got it set up. And yeah, and the PC 3 and 4 also match expectations. So yeah, so we can use these things, right? They're actually like, they can get like really stellar results that nobody could see with them. But there's some other stuff happening in the background. So you'll notice that all this stuff was pretty much focused on the case where I somehow get the graph, and then we have short reads and do stuff with it. But things are changing, and now we have complete whole genome assemblies. I want to explain a little bit about why. I guess we're at, I'm going to do the explanation and we'll take a break. I think that makes sense. So the explanation of why is we have single molecule sequencing. And this is really a quantum leap in every sense, in what it allows us to do in biology, bioinformatics. This is called third generation because we had the first generation being Sanger sequencing, the second generation being short read sequencing, often using fluorescence imaging and these kind of ensemble observations of many molecules. And the third generation is single molecule. And how does that work? So this is the first, I guess, the order of presenting these two topics it's kind of irrelevant though. First and second don't really matter. Basically in the mid 2000s, like a bit before 2010, this company, Pacific Biosciences, which is always called PacBio, I think it might even be officially PacBio now, they come out with this really ingenious scheme. So they make a flow cell in which the bottom is glass, or it's a transparent medium, and there are little wells, really, really, really tiny wells that have been put into I guess a photoresistive substrate on top of it. So you see that in A, right? So there's this is glass. Then you have this, I don't know what the material is. It may be silicon dioxide, but basically they've deposited it and they've got these little micro wells drilled in it. And what they realized is that there's this evanescent wave that would come into this region a very, very small distance based on the size of this well, the diameter of it, and the fluorescence and the frequency of the light. And this allows them to have a detection area for fluorescent imaging that is really, really small. It's like a picoliter. And it can encompass pretty much just a polymerase and a single-stranded DNA molecule that which it's synthesizing the second strand of. So that's the start. And the way you can get signal out of this is through fluorescence. And the way that works is that instead of regular DNTPs, in the solution, you have DNTPs where you have a polyphosphate linker and then a fluorophore. And so the polymerase can work with this, like, it's crazy multi-phosphate linker. It'll cleave that, and it'll use the energy from that bond to actually drive the polymerization of the second strand. But as it's cleaving it, it's going to hold onto that fluorophore, and it's going to be really, really close to this detection area. It's going to be inside of this zero mode waveguide system. And when that happens, high frequency fluorescence imaging will detect a peak associated with a particular fluorophore. Each one is associated with a particular DNA base. And so this allows for the observation of the progression of a single polymerase. So these fluorophores are going to be, diffusing in and out and, moving through Brownian motion in and out of this area. But what's detected is that they actually stay in that region much, much longer than you'd expect by chance when the polymerase is interacting with them. And so then software, I think this is really an idealistic image, but basically this is what the software is seeing. It can say, oh, hey, I see an A being incorporated because my fluorescence signal in that well for the A fluorophore is really high, right? And so it progresses like this. And this is really amazing, but for a long time, people didn't quite know how to use it well because the error rate is super duper high. This example at the bottom is just not, it's not really like that. This is very idealistic. Typically what's happening is you've got an error for, every 10 base pairs, you're gonna have an error. And you often have insertion errors because basically the fluorophore comes inside the well, and it looks like a base got inserted, and so there's these very high insertion errors, and then there also can be problems with the secondary structures of the molecules, but those are really more minor. The real problem is the error rate. And so the genius move was like, hey, we can just sequence the same molecule of DNA over and over. And the amazing thing about this error processes in PacBio is that they're super uniform, almost like white noise. And so if you keep observing the same thing over and over, you can build up a consensus of what the bases should be, even if you have really high rates of error, because the errors don't recur in the same context. And so they have this kind of protocol. You have double-stranded DNA, you get these two hairpin kind of adapters, and bind the DNA polymerase that gets loaded onto this thing. Some of these systems end up with polymerase down in the well that don't know exactly how they do that. Might be kind of company secret. Then you do rolling circle amplification, and the same process is being observed. And this is really transformative. And I'll get to why in a second, why this particular aspect is so important. So the other thing that's happened, the other technology that's now in play is called nanopore sequencing. And this is postulated by a bunch of people from the 1980s and 1990s. It's an idea that's kind of latent, and Oxford Nanopore is the current world leader in this because they have all the IP on it. Can't wait for that to expire. I love them, but it'd be great to have some competition in this market. Basically, the idea is you can measure electrical changes caused by DNA being pulled through a nanopore that is itself embedded in electrophoretic membrane. So there's a voltage across this that is driving, it will drive DNA through the pore. And this is also, to help be more progressive, there's also helicase involved that makes sure we have a single strand of DNA going through that pore. And so the idea is, this is flowing through, this DNA is flowing through. As that happens, the different bases have different electro biological entities that can be observed with changes in electrical current, and that results in base calls. This is not really what it looks like. I got some real data to show you. This is really what the signals look like. They're absolutely difficult to work with, but they do show very characteristic patterns. It's just not like one-to-one correspondence between like a current and a particular DNA base. Actually, there's a bunch of DNA bases inside of this at a time, 5s, 10s, something like that. And it's like that kind of Kmer that you're observing. And then there's time dependence too. So this whole signal is like shifting around a little bit in sort of funny ways. And so it takes some pretty good machine learning methods to extract base calls from it. And Problematically, the error profiles are not uniform like in nanopore, so you will make the same error at the same place over and over and over. And this is getting better, but it still happens enough that it's difficult. So reads coming out of the nanopore sequencing, like basically because the touch is so light, like there's almost no input into the system. Like the PacBio system, you get photo bleaching, it will break your polymerase eventually because you're shining these bright lights on it. This is not the case here. Here, you can really sequence a whole DNA molecule. You're basically limited by your library preparation. And so 100 KB reads are easy to get. It's possible to get mega base pair reads. Those are really valuable for genomics. But the problem is it has this kind of intrinsic high error rate. That's coming down, and it's now around 2%. But it was as high as 15% previously. So why is this like so important? Why does this stuff matter? Because you should be like, well, I can sequence DNA with a short read sequencer. Why do I need these long reads? They matter because genomes aren't random. You know, if genomes were high entropy sequences, 250 base pair read would be able to access every single part of it. That's not how life works. Life works by copying. And copying leads to repetitions in the genome sequences. And those repetitions are what makes it impossible for us to assemble short reads. So this is from Gene Meyer's paper on what's called the assembly string graph, which is the basic formalism we use when we assemble genomes. So the gist of it is this: if you have repeats, which are these colored arrows in a genome, and your reads fit inside of those, if you make a graph out of all that stuff using what's called overlap consensus and overlap layout consensus, so you kind of like aligning all to all and then making a graph, you're going to expect a structure that collapses the repeats. Because once something goes inside of this, you can't tell if it comes from the first or the second black arrow, first, second, or third one here. We don't have this genome. That's a truth we're trying to get access to. We just have the string graph. What happens is that until your error rate is lower than the differences, so there will be differences between these sequences, right? But until your error rate of your reads is lower, or kind of at the same time, the read length is long enough to actually span through these things, then you can't resolve this into a clear assembly. And so PacBio HiFi is basically giving us the low error rate. The read lengths are actually long enough in practice, about 10, 15 KB, to span most repeats in the human genome and in a lot of eukaryotic genomes. Nanopore, though, takes it to the next level because the reads can be really, really long. And so combining these two actually lets us to unfold the string graph. And this is practical results from the past year that we can now automatically take the sequencing data and get a diploid assembly of a single human being. And it seems to work in most species that are being tried on. And not only do we get an assembly, the accuracy is really incredible. You get one error in 100,000, and that's a starting threshold. Some of these assemblies have higher than QD60. It depends on how much data you're putting in, obviously. The process, I just go quickly through it because it's an interesting case where the ideas about pangenomics are actually feeding into assembly concepts. So basically, you can build this kind of initial graph using really, really long k-mers. Like, so you're doing overlaps between really long k-mers, like thousands of base pairs, hundreds of base pairs. And that has a structure that's sort of suggestive already of the actual underlying genome. But what's then done is that really long reads from nanopore, so this is built from HiFi data. It's really low error, but not really long. Then really long reads are used to disentangle funny stuff in that. And this is actually done with a tool that was made for pangenomics called Graph Aligner, which aligns sequences to graphs. And so then there's this process that is then, additional markers can be used to actually get annotation of what the paternal and maternal components are. Those are typically based either on trio data, you actually have the parents handy, or you can use something about proximity in the cell, like Hi-C. And that'll actually allow you to say, well, these bits tend to be closer together physically. The bits in gray, there's not enough differences between them to separate them, but we can kind of infer that there must be two copies, one in each haplotype. And so this results in this complete assembly. This is an example of it in the context of this HD002 sample. So they're able to resolve almost everything automatically end to end with only a few cases where there's ambiguity. And And you'll notice two things, which I'm going to talk about after the break. So one is the sex chromosomes. They're stuck together by their pseudo-autosomal region. The acrocentrics, they're stuck together by ribosomal DNA, but there's more to that story. So I say we'll leave it here for 5 minutes and come back. Is that good? Are there any questions so far we want to get to?
I'm just curious to know that what formats is used to store these genome charts? How does that look like?
What?
So I can repeat. The question is about what kind of file format do you use to store these graphs?
Yeah, there's a format that is called GFA, Graphical Fragment Assembly. It was developed for assembly problems, and it also will work for pangenomes. And there are several proposed ways of doing that, several methods that are pretty much equivalent. They just they take the kind of assembly graph and they add the path information from genomes that are put into the graph, into it. It's a text file, by the way.
And another question is on the read aligners. So those kind of like do some backtracking steps, try and find the most optimal alignment, but just go to H.
There's not really backtracking. You typically just do you attempt multiple contexts where you think that the mapping can go, and then compare the actual base level alignment quality between them. In fact, the quality of the alignment of the whole read pair will typically be compared. The backtracking makes sense in the context of BWA align and bowtie. So those are working on a structure called the FM index. That is like a full text index of the reference. We use similar structures in VG map, but we don't use backtracking because backtracking has exponential cost in the number of errors that you can consider, and or differences, not errors, differences relative to the text you're mapping against, and makes, because of that, you can't look at indels easily, because indels, the number of possible indels is very large, so you can only look for very, very small numbers of them. It's not really a practical method. I know it gets used in the context of ancient DNA still, but that's really because the reads are very short, and you don't have much hope of looking for indels, I think.
Yeah, just extra information. FM index referring here is based on BWA. So if you think of BWA, which is taught on Tuesday, it's used the backtracking. So that's why it's like there.
BWA align, yeah. We based on top of BWA mem. So the FM index was developed by actually collaborators of Luca and I. Yeah, smiling, a friend. And then that was implemented in BWA, is used to provide this full text searching. The first versions of that use backtracking. As the read lengths became longer and the error rates kind of got better, it was no longer sensible to do that. And so BWA mem, it finds what are called maximal exact matches, which can be done in linear time by comparing a read or sequence to the FM index. And then those are chained together, actually. And then the final alignment can be done with a pairwise alignment algorithm that works at base level. And like actually goes to the place you think that the read maps and looks at it. That same FM index concept is generalized into something that works on graphs in VG map. So in fact, VG map is like a pretty straightforward generalization of BWMM to work on graphs. That's the whole design of it.
Any other questions? Also, people on Zoom, feel free to unmute yourself. All right, looks like no questions. Oh, yeah, sure. You want to come here? So I think it's easier.
Yeah, it's a little easier here.
Hi, so I have a question about Verkle, because you showed in your VG paper that Graph Aligner actually has worse performance than the VG method, and Verkle is using the whole pipeline, so you can easily modify each step. So,
Yeah.
and Verkle has been used mainly for assemble the newest deployed genome HG002. So have you thought about kind of change Graph Aligner into?
Well, yeah, it would be great. Graph Aligner is sort of a beautiful nightmare. It works. decently well. It's optimized more for long nanopore reads and long sequences. And I think that's part of why it doesn't work well in the short reads that we're testing it with in this other case. So you're absolutely right. It doesn't work well in short reads. For longer reads, I think it begins to become okay. It has some technical limitations that make it the base level alignment not super good. And the specific issue is it uses what's called an edit distance alignment. So it weights mismatches and gaps exactly the same. A 1 base pair gap is like 1 base pair mismatch. A 2 base pair gap is like a 2 base pair mismatch, and so on. In real biological systems, you don't typically see that kind of relationship. Usually, there's a sort of power law distribution, a long tail distribution of indel lengths, and so that's why we use this thing called affine gap alignment. The effect of that is that you can't really use the base level alignments of graph aligner for much downstream, but you can use what's being used here is actually very high level. It's not about the base level alignments as much as it is about alignments across node sets that are very big. So graph aligner can do a good job of finding like the alignment between the 100 KB nanopore read and a 10 KB node, right? It can be very confident. Okay, it should map through that node, even if the precise way the bases are lined up is not accurate. Is that helpful? And yeah, I think you're right. It'd be amazing to have a newer, better method that was just in general a better aligner and maybe faster too. But graph aligner is what we have right now, so it's an open problem.
Yeah, right now, like the state of art popular assemblers, Verkle is the slowest. It takes about 5 days to assemble the HG002 hi-fi reads.
It does, yeah.
Just like 2 to 3 days, which is manageable.
Yeah, although, actually, this is a quote directly from Gene Myers that you don't care, in the end, you don't care about being fast if you're wrong. Like, and actually, we're looking at structural variable loci in, we're looking at both Verco and HiFi-ASM with the ultra-long integration. Verco is perfectly accurate on all the ones we've looked at, using cross-validation with short reads, and those have been cross-validated with ddPCR, so we're totally confident in it. It's not the case for HiFi-ASM, at least today. It could just be a matter of algorithmic tweaks, but I think it might be something kind of deep, like the fact it's actually doing the alignment of the reads to the De Bruijn graph, or the ULA graph as it's called. Like that's not exactly equivalent to what's being done in HiFi-ASM. So we'll see how that evolves. But I think you're right to point out that it's very slow and it's frustrating. But I want to be, I'll pay twice as much for my assembly if it's perfect. Because it's going to cost me a whole lot if it isn't.
Okay, thank you so much.
Yeah, thank you. Great questions. Ooh. Okay, so I guess we pause for 5 minutes. So, yeah.
Yes.
And we're maybe running a little low on time, but I think it'll work out because the last part is more exposition of applications.
Okay,
cool.
Sounds great. Thank
Great.
you.
Okay, I'll just in 5 minutes come back. 5-10 minutes.
Yes, perfect.
5-6 minutes. Okay, cool. Okay, I'll get into it.
Yes.
Thanks, Luca. I guess people are filtering back from the break, but I'm going to start for the interest of time and hopefully not rush too quickly. The last part of this is all about applications of these ideas once we have complete genome assemblies, like the ones that I'm talking about being based on nanopore and HiFi. So then the question, this is a question that has motivated my research for a long time, which is like, in the future, pretty sure we're going to have lots and lots of complete assemblies. So what's the complement to that future abundance? Tools to understand variation between many assemblies at the same time. And so I think I probably convinced you that variation graphs are a nice foundation for that. But the issue is like, how do you make the variation graph from the whole genome assemblies? So it's kind of daunting. So there are methods. I apologize for not including these because this is a lecture for a course, and so probably it's interesting to learn about them. But there are several methods. I'm only going to talk about the one I've been working on, but I'll mention the others right now. So the first one that was published for working with whole genome assemblies and doing this kind of multi-sample variant calling graph building is called minigraph. It's by Hung Lee, who's the author of BWMM and Minimap2 and a bunch of other cool things. And that works by taking a reference and then in order layering new sequences onto that, looking for bits that aren't already in the graph. And so it enforces a global order to all the sequences you put in that's based on the original reference. And so you can layer stuff up and you find the variants and play the same game. You make a graph just like the ones I've been showing before. Then that process only can see large variants, more than about 50 base pairs by its design. To then refine that and add small variation on top, Benedict Patton, who's also a longtime collaborator, and has worked on comparative genomics applications of these ideas. He and his group, including Glenn Hickey and others, they developed a way to add the multiple sequence alignment information on top of the minigraph. And that's something called minigraph cactus. So both of those methods, they start with a reference and they add stuff to it. And the particular order that you use, the particular reference you choose, affects the shape of the graph. I was interested in, and remain interested in, building unbiased methods to work with collections of genomes. And so that's what the Pangenome Graph Builder is. It's an attempt to make an unbiased approach that actually starts from an all-to-all comparison and alignment of the input genomes. And the idea being that if we do all-to-all, we're not order biased. Every reference we put in, multiple references, multiple haplotypes, they're all kind of equivalent. They're treated equally. And that makes a lot of the downstream work and inference on top of that much simpler because we have those kind of key guarantees. So the way that this works is that first we had to make an entirely new kind of aligner because the existing approaches were not scaling to aligning like whole chromosomes to each other, or worse, many whole chromosomes to each other. And that's this tool that's called WF-MASH. It's based on an algorithm called BiWFA, which I won't have a chance to go too much into, but it's pretty amazing. In recent improvement in a fundamental bioinformatics problem of pairwise alignment. So then once we have these alignments, that we can convert those into a graph with an algorithm that is pretty much completely described on this one figure. I'll get into that in a second. That gives us this graph on the bottom that is just kind of an exact representation of all the information in the alignments we have made. And then that graph can get pretty crazy, and it doesn't really expose the latent structure of the pangenome. And we have different ways to pull that out, to organize the graph, and then to apply multiple sequence type alignment algorithms to refine the local structure, to make consensus graph, and to make an improved version of it. So the initial alignments, as I said, they're made by this thing called WF-MASH, and that's linking an algorithm called MASH-MAP, which is a very fast homology search algorithm. It can basically you can map a whole human genome to another one in a few minutes with this. But it gives you very approximate high-level interpretation of that. It says like, this 100 kb segment maps to that 100 kb segment. It doesn't tell you the base level alignments. So we wanted to take those homologies and then turn them into precise alignments. To do that, we cannot use something like standard dynamic programming. This one on the left here is Needleman-Wunsch. This is showing you the score that you get of a given alignment reaching to a given place in the matrix. And it goes like the ones that are in red would be very unlikely because they're high scores. The ones in 0, the lower ones, are more likely because they're lower. But you see that you have to fill out this whole matrix. I cannot do this if I have 100 copies of chromosome 8 against 100 copies of chromosome 8. It's exascale. So obviously that doesn't work. We use this homology approach to reduce the scale, but still, if we're thinking about hundreds of kb versus 100 kb, it's completely unimaginably pointless to fill this whole thing out. And what a researcher named Santiago Marcosola, who's in Barcelona, realized that you can actually progressively explore this matrix in such a way that you're tracking a wavefront of best scores. And if this continues until you get to the last cell in the matrix, you found the optimal alignment in that set that you've explored. So he's developed this, and then we extended it into something called high order WFA. Basically, it's aligning not single base pairs, but chunks of sequence that are kind of the size of these chunks of sequence, tens to hundreds of base pairs. And this is looking at the alignment of 2 mega base pair size regions of 2 E. coli genomes. And basically showing us in gray the parts we explore, and green are the parts that we find matches in. And so we do this in a kind of complicated alignment algorithm, but nevertheless very efficient, allows us to make the alignments. And then this graph construction is actually very, very simple conceptually. So the alignments are just telling us like, this character in this text is the same as this other character in this other text, and so on. So if we were to represent those as these dashed lines between the sequences, then we do this kind of transitive closure or union find operation to collect all of the ones that map together. And then those get converted into a single base in the output graph. And we do this and remember where the paths fit into that, and we can make a variation graph. And then there's this process of normalization. So the graph starts out very disordered. We actually learn using a kind of gradient descent algorithm that's run in parallel, a organization in one dimension that kind of is minimizing the distances traversed by individual paths through the order. So it's kind of finding this modal representation of all the genomes put together in the graph. And then locally over that, we can run MSA algorithms to reduce the complexity of this representation. This is like a very underaligned, messy graph. If we were to run this process, then we would end up with something where every base is compactly aligned to other ones that can be aligned against. And so then downstream of this, we have a whole toolkit that's, I guess, it's kind of like SAM tools or something, or BED tools, but designed to work on these genome graphs. You know, it reads these GFA files and then lets us look inside of them. And we can do things like get a visualization in 1 dimension or 2 dimensions. These at the top are 2-dimensional layouts. This particular example is from this paper on AUGI, it's called, that zooms in on the MHC, which is in A. It zooms in on a gene called C4, or 2 genes called C4A and C4B. And this is a copy number polymorphism, a VNTR, variable number of tandem repeats, and basically different haplotypes have different counts of copies of the genes. And these provide visualizations to see that. And we can zoom in and look at it, or we can extract the pairwise alignments back out of the graph, kind of using all the information of the graph, but then looking at the pairwise relationship between a given reference and a given target. And yes, okay, I thought I had another slide here, but it's actually later. Okay, so that's like the tool set. We can, and then the data we have is just awesome. It's really fun. The Human Pangenome Reference Consortium, which I'm a part of, it put forward this project to make hundreds of reference quality assemblies of human genomes that are from cell lines, and they're haplotype resolved, and they're really cool. I won't get into the assembly because I talked a bit about how we want to do that, but we have an assembly process that's pretty good. And then we built multiple versions of the graphs using these other methods, which I mentioned, and PGGB. And this was a little bit due to competition, a little bit due to the fact that the properties of the different methods are interesting to the authors of them. You know, they're not all the same. But it's also because of the importance of cross-validation. So we now have like 3 sets of variant calls we can compare to each other and see how well we do. And so there's actually been a preprint on this. It's out. It's been accepted for publication. We're looking forward to pushing that forward. So this is just quality evaluation. So we can make the graph, and a decomposition of the graph allows us to look at a VCF file that has variants in it. And we can compare those to another VCF file with variants in it, representing some truth set that we've established. And so these give us these precision and recall features for variants detected in the 2 base level graph pipelines. And we see that for easy stuff that are SNPs, it's just, we're perfectly in agreement, get exactly the same kind of quality. This is approaching the quality of the actual underlying assemblies in terms of error rate of SNP calling. In regions that are more difficult, like regions that are repetitive in the reference, tandem repeats, then there are kind of specific differences between the 2 methods that show up, and in general, we don't do as well. But that's to be expected. Also, given that we have to compare to other samples in the frame of the reference. So we're still reference biased in this evaluation. And this also works for structural variants. And also when the structural variants are easy in easy regions, so just a big indel, we're almost as good as for SNPs. And these are, again, the two methods. We can see the precision and recall curves at the top that were pretty good across 10,000 base pairs plus or minus. So we can accurately detect very large structural variants inside of the graph we built from the assemblies. And so these then let us look at really interesting medically relevant loci, often involved in immune response, often places where there's diversifying selection going on, and there's many haplotypes in the population. In A is this is a view of the Rh blood group system locus, and in the mini-graph cactus graph is used to provide this. And then this is kind of a manual description of the way that the different genes are seen across different haplotypes that have been extracted out below. And then this is in MHC. There are HLA-A and many pseudogenes of it nearby, including HLA-Y, I believe. And this is kind of cool because this region of HLA-Y, it's highly diverged and between the reference and a lot of the samples. In fact, this is one of the largest regions in the pangenome that is genic, or in a euchromatic region, and is not present in the reference. And so it actually is an ancestral state that we have. That's not in the reference. The reference is a deletion of that state. So, and then we also can look at a whole chromosome graph and get some kind of sense of the high level architecture and variation patterns. So these are the 2D visualizations that are optimized to minimize path distance, and they're run across. We do them for all the chromosomes, but here we're just looking at a few that are kind of interesting. Chromosome 1, you have a very typical pattern. The euchromatic arms are just like wires. If you zoom in, they're little bubbles that represent SNPs and small variants, but in general, it's all pretty clean. The centromeres are very complex and have very high rates of polymorphism, and they're very hard to align properly. And so in these graphs, you see them as these big hairballs. But in effect, they are VNTRs, variable number of tandem repeats of repeat units that are have functional significance for the behavior of the centromere. And so those usually collapse in some kind of way, become very tangled and complicated. And yeah, so then we have 8 and 14 is acrocentric. I'll talk a little bit about that later. And you also see a lot of variability near the centromere in the pericentromeric regions and in the subtelomeric regions. This is an example in 14. This is actually the immunoglobulin heavy chain locus, which is near the telomere. So then just zooming in on this weird loop in chromosome 8. This is a 5 megabase pair polymorphic inversion. It is flanked by clusters of things called beta defensin genes, which are basically antibiotic genes involved in innate immunity. And so it's fascinating that those are in these repetitive sequences that are on the edges of this inversion. This is showing the two references relative to each other. They actually have the inversion between each other. The CHM13, I'll talk about it in a minute. And then the regional, we can see in the graph, because we're really confident in the variant calls, we can see in the graph the variant density. In fact, around this whole region, it's higher than everywhere else in that chromosome and almost everywhere else that you see in the entire genome. So it kind of shows like these polymorphic inversions, these are actually flipping at a very high rate. It's shown in other papers recently. Those should suppress recombination. They should increase the effective population size. And there's sort of must be some kind of selective pressure that drives immune genes to be closer to these kinds of things. And so this is looking at the loop itself with gene annotations on top of it. The region where the breakpoints are occurring is down here in this tangle. And this is actually an interactive visualization. We can zoom in and look at these kinds of things. And this is this particular cassette that's traversed multiple times in its variable number in different haplotypes. It has these DEF-B defensin genes on it. And something really fascinating, there's this one down here, it says FAM90A. This is a gene of unknown function that has incredible polymorphism in copy number. Some haplotypes have just a few copies, some have 100, Yeah, this is the MHC. I'm gonna go a little quick through here just to move on because I want to show you some of the final results from a particular study we did with this data. I showed this kind of figure before. There's a C4A. We zoom in on it. Then C4A and B are collapsed. Basically what's happening is that there's this almost exact repeat in a lot of these haplotypes. And the collapse actually occurs. We thought it was an alignment problem. Like you shouldn't get a bubble that's a loop. You should get like separate SVs, That was what we expected to happen. In fact, when we build this graph and then extract the pairwise alignment information out, we can see that what's happening is that you have a reordering of the sequences in different haplotypes. For example, you don't always have C4a and C4b. That's being shown over here. So usually C4a, C4b is like red, green. Sometimes you have C4b, then C4a. And so this is this kind of nonlinear pattern of evolution that's not expected, or wasn't really clearly expected before, but we can see it very precisely now. It's probably modulated by interlocus gene conversion or interallelic copy gene conversion that's driving these kinds of effects, and or homoplasy. All these things might be playing a role. And so we see that the segmental duplications and the structural variations have this kind of thing happening. And there's a really cool companion paper on that topic. So then I'll discuss the companion paper I've led in these last minutes of the course. And it starts with the complete assembly of a human genome, which I mentioned in passing. It's called CHM13. And this schematic shows across the chromosomes, all these regions in black, there were regions that were ends in the reference or not assembled correctly in the GRC reference. And so we recovered 8% of the human genome that was not assembled, including the acrocentric short arms. These were quite literally ends and just inaccessible to any kind of study. You might have noticed before on the HG002 assembly graph, all the acrocentrics come together. This is also true in the CHM13 assembly graph. And so that's what we're focusing on here. This particular pattern is really funky because every other chromosome managed to be separate, with the exception of X and Y, which often come together in the PAR. The fact that these are always together in the assembly graphs, and kind of suggested that something was going on. Then we looked in the HPRC assemblies, the Pangenome Project assemblies, and there's this misjoin identification, it's called, this quality control thing we did. Hung Lee used alignment to find cases where contigs appeared to map to multiple T to T chromosomes. The particular graph, every node is a contig in some assembly or one of the references chromosomes for the acrocentrics. And almost all of these misjoins are happening between different acrocentrics. And so I wanted to take this to a higher level and look at everything all together to exclude the possibility that there were other things we weren't seeing. And so we do an all-to-all mapping of all the HPRC contigs. So we're mapping these 90 haplotypes, whole genome, all to each other. And we keep contigs greater than 1 megabase pair. For what it's worth, they're almost all greater than 1 megabase pair. So that's not too much of a filter. And this particular region, so the sex chromosomes are kind of together. That's not surprising. But then we see the same kind of characteristic pattern with the acrocentrics. Here we're labeling the contigs by which chromosome we think they're from. And we get this sort of muddly mess in the middle of this. This is a two-dimensional kind of graph layout of this particular type. The fact that these are laid together suggests that they have a lot of connections between each other. And we then, to quantify this, apply a community detection algorithm to this kind of graph. And you get single chromosome communities or chromosome arms, like chromosome 3, it's in 2 pieces here, with the exception of the sex chromosomes. Here, chromosome X component 1 includes Y as well, and the acrocentrics. And there are only really a handful of other cases that don't fit into that pattern. And so that's really suggestive. This is coloring by the communities that are assigned. And you see that there's this one kind of giant community that's encompassing most of the acrocentrics. And there's also another one in the sex chromosomes. I don't have time to talk about that, but there's a nice supplementary figure in our paper on it. So then we said, okay, this is confusing. We want to say this contig does come from that chromosome. In order to do that, we have to have some high confident region that kind of anchors it to a particular chromosome. And so we extracted contigs that were mapping across the centromeres of the acrocentrics by at least a megabase pair. And we assigned them to a chromosome based on their Q-arm relationships to the T to T. Then we build a graph with PG to V from everything. And this is what we get. So now this is a base level resolution of the homology relationships in the whole set. And we're coloring here by where the T to T CHM13 assembly is. And so you have 14, 22, 15, 13, 21. All these Q arms are basically isolated. As we get close to the centromeres, there starts to be patterns where the graph is actually collapsing together, like both of these come together. That happens because of homologies in the underlying sequences and typically is indicative of recombination type processes. This begins near and a little bit before the centromeres in 13 and 21, 14 and 22. And 15 is a little bit of an outlier, which isn't surprising if we look back here. In this kind of all-to-all comparison, it also is an outlier. So yeah, and then zooming in a bit more, I'll get into this, but there's some particular regions that are really interesting. These are basically copies that are one-to-one copies on multiple chromosomes, but at very high identity. And to understand these, we do this process we call untangling, which in short is extracting alignments out of the graph. And basically we segment the graph. We have a query. So this black line, we want to ask which of the acrocentrics in this case it's most similar to in this region. And so we can do an estimate of Jacquard between all the possible targets, and we can rank them, right? So get a multi-mapping, in fact. It says this is the goodness of the matching, and this is the rank for all these different alternatives. And then we do this funky thing that is probably hard to understand in the context of this talk, but basically we can say, extract the coordinates on a particular chromosome for this. Like even if the best mapping is the 13, let's say our contig is from 14, we can ask where on 14 would that occur? And we use this to make these crazy plots that show us the pattern of relationships as if we're kind of walking through this graph. So here, we're imagining we're walking down chromosome 13. We get to about here, and that's where this figure starts. So here, everyone is like completely the same, with the exception of some SVs, I guess, relative to chromosome 13. But then these contigs, as you go through the centromere, in this region between the ribosomal DNA and the centromere, and by the way, I didn't describe this clearly. Acrocentric means the centromere is close to the telomere on one side, and in humans, all the acrocentrics have the ribosomal DNA repeats on them. So that's the biology driving this. They have similar function. And so in this region here, you have a lot of long non-coding RNA type genes and stuff. And it's not completely like satellite sequences. It's not just repetitive centromeric type stuff. There's material here that must have functional significance, and it's not evolving in such a way, in like a satellite type way. But there is a satellite sequence in the middle called SST1. Right? And so this pattern here, it's like saying this contig, it looks like 13, and then it looks like 21, then it looks like 13, and so on. This one looks like 13, then it looks like 14, right? And so we can quantify the pattern, we can add up the kind of matching against different targets and extract different entropy metrics from this. And these in particular, we use them, this one here, to derive what we call pseudo-homologous regions. And so the places where the entropy goes up are places where we think there's recombination. And these sum to about 18 megabase pairs, and they're probably an underestimate of what's happening. And then finally, we did a scan for PRDM9, which is it actually modulates homologous recombination during meiosis, and find that in the middle of this region where we see all this activity, there's this peak that corresponds to the array repeat SST1. There actually are targets for PRDM9 in that, and that might be actually modulating this kind of process. So I'm just going to run through these quick just to show you, these are just in different chromosomes, you say the same kind of pattern from a different point of view, right? Now we're looking from the point of view of 14, it's mixed a bit with 22, and then it mixes a bit with 13 and 21, and 15, a little bit less is going on, but as you get close to rDNA, again, you see the same kind of pattern, and we think that maybe there's this large satellite sequence that doesn't recombine as actively on 15. And here's 21, again, looking back the other direction, you see the same structure we saw on 13 and 14, and 22, okay. Just another one for the pile. And then the really fascinating thing beyond this is that if you look at the SNP variation inside of these regions, it suggests a faster decay of linkage disequilibrium, which would be correlated with higher recombination rates or potentially a higher effective population size. And again, I'm running out of time, so I won't dig into this. Yeah, I'm done.
The room, like they may need the room lighter. So
yeah, we're gonna finish here. So this is the last slide. So this is basically just to drive home the findings here are basically that there's this pseudomologous regions, which we're pointing at. There's this SST arrays in the middle of them. They're evolving under concerted evolution, so they're all mixing together. That's what this phylogeny of the repeat units shows, that all the different acrocentric chromosomes, they're mixing together. Other repeat units that are similar in the genome, they don't do that. And this is here showing this kind of case. And then this is a multiple untangling, so it's like a multi-mapping, not quite like the ones I was showing. And this checkerboard pattern specifically is what is indicative of recombination, right? And then we relate this to some existing results using FISH. They look at Robertsonian translocations. Those are translocations where two chromosomes come together, they make a dicentric chromosome. It's quite large, and then the other material is lost. They use FISH to show that these clones on 14 and 21 are in the Robertsonian, and these ones in red aren't. And that gives them a breakpoint, but they didn't have the assembly of the region to look at. We can show that these actually are consistent with recombination between non-homologs in that region. And that gives a kind of solution to Robertsonian translocations. Anyway, I'll share the slides so you can look at these citations, but this is like, there's a lot of work basically building up to this that we're confirming. This is even from the 70s, and you see different acrocentric chromosomes hanging out in the same nucleolus. Here it's 14, it's 13, and 21. And it's really surprising that we're able to see that with this new data. And also I want to point out that because we do a population genetic study on these graphs, we avoid some of the problems that were had when trying to look for these associations. Here, people did hundreds and hundreds of FISH experiments to try to see if 14 and 21 recombine. And they were not really able to make a conclusive result. They find one instance in hundreds of cases, which is consistent with this picture that there's recombination exchange happening between these pseudohomologous regions. They're on different acrocentrics, they're physically proximal, and that facilitates the recombination. Usually that should be gene conversion. Sometimes, maybe 10% of the cases, there'll be crossover type recombination. If that occurs between 14, 21, and 13, then the inversion of a certain region on 14 can trigger the Robertsonian translocation. And so that is basically it. And thanks, everyone. Thanks for going a few minutes long as well.
Thank you so much. This was fantastic. And yeah, if you don't mind to share the slides, because there are so many important references here so people can dig more. But yeah, thank